Hello friends, welcome to Bunk once again, and today's topic is urine formation. Okay, so this is basically the physiology of urine formation. So basically there are three steps. We can see glomerular filtration, tubular reabsorption, and the last one is tubular secretion. Okay, so what is happening in glomerular filtration is like the pressure, the diameter of this afferent, you can acknowledge the diameter of afferent is more as compared to the efferent one. So what will happen when the input is more and output is less? So the pressure will created in this region. This region is what? This is glomerular. Okay, this one is glomerular capillaries. Glomerular capillaries. Okay, this one is glomerulus, and here the when the pressure is created, the level of glomerular capillaries, what will happen? This in the filtration will occur in Bowman capsule. Okay, so this type of filtration, because of the difference of afferent and efferent, and then the pressure is created the level of glomerulus. This is called glomerular filtration, or you can say it ultrafiltration. Ultrafiltration. Okay, so it can also be called ultrafiltration, right? Now, let us see what is tubular secretion. So, whatever filtration is occurring, it is coming inside the Bowman capsule. When it is coming inside Bowman capsule, it is called GF, glomerular filtrate, or you can say UF, that is ultrafiltrate. So, what else a glomerular filtrate is lacking, or what ultrafiltrate is lacking, is the plasma protein and corpuscles we have discussed in the last video. Okay, so let us see what happens when it goes further. So here in glomerular filtrate, what happens, like protein, which is an important substance, and amino acid is of course the smaller form of protein. So amino acid, protein, you can say glucose, you can say the smallest form of carbohydrate. So these are the important substances which need not to be eliminated out of the body. So what happened? It is getting reabsorbed in this peritubular capillaries, you can acknowledge. Okay, so in peritubular capillaries, these important substances such as water, glucose, amino acid, and these needed to be given back to the circulation. So how it is given back to the circulation? It is given by selective reabsorption process. Okay, so tubular reabsorption also called selective reabsorption. So selectively it is taking the thing, okay, selectively it is taking the thing which is required for the body. So which one is glucose, protein, and amino acids, and given back to the circulation via peritubular capillaries. Okay, and then during by the course of filtration, or by the course of tubular like by the course of selective reabsorption, what happens? Sometimes the substances which need not to be given to the circulation, rather it has to be completely discarded out of the body. Those are toxins. Okay, so toxin waste should be completely discarded out of the body. That is H plus, like H plus ion, and you can say K plus ion. Drugs, drugs need to be completely discarded out of the body once it is used, and you can say creatinine, and you can say parahyperic acid. Okay, so these are the toxic waste. Toxic waste should be completely discarded out of the body and not at all absorbed. So these substances are what? From peritubular capillaries, which are somehow bypassed from the afferent to efferent and not at all given into the ultrafiltrate for urine. Okay, so what will happen in that case? In this peritubular capillaries, these substances are flowing which can going into the circulation, right? So what will happen? The C part you can acknowledge here. This tubular secretion is occurring. So tubular secretion is doing what? The creatinine, the drugs, the important, the parahyperic acid, the waste product, which should not be given to circulation, rather completely discarded out of the body, is given back to the capillaries of nephron. Okay, so that it should be discarded out in urine. So these are the three steps for urine formation, which is glomerular filtration, selective reabsorption, and tubular secretion. It's a very simple thing. In ultrafiltration thing, the diameter of afferent and efferent is variable. Okay, so afferent diameter is more, so input is more and output is less. So what will happen? The pressure will be created at the region of glomerulus, and then filtration is occurring, and filtrate is going inside. It's coming inside glomerular capillaries. Okay, you can see this, not glomerular, basically it's coming inside this Bowman capsule. You can acknowledge this thing. So this one is Bowman capsule. So it's coming inside Bowman capsule, and the filter rate is called glomerular filter rate or ultrafiltrate because glomerulus is basically filtering it. Okay, but during this filtration process, the substances which need to be given back to the circulation, like glucose, protein, amino acid, is the essential thing that is given back to the circulation. So how it will be given back to the circulation? It should be given back to circulation by by the circulation by this peritubular capillaries by the process of selective reabsorption. Okay, so now I hope you understand what is happening. So in this thing, you can see this is afferent, and this one is glomerulus, and this one is the smaller diameter one. This one is efferent. Here you can acknowledge this Bowman capsule, like the thing is coming like this. Okay, and the branch of efferent artery, it is called peritubular capillaries, which is running parallel to it, so that substances need to be excreted and eliminated like this. Okay, so here, due to the afferent and efferent diameter variation, what will happen? Pressure will be created in this glomerular duct of capillaries, and then filtration is coming like this. This is called glomerular filtrate or ultrafiltrate. Okay, but during the course of filtration, the things like here, things like amino acid and protein, and you can say water also, but basically glucose and all these things. Okay, so these are actively transported, like with the help of ATP expenditure, it is given to this peritubular capillaries. Say it is peritubular capillaries. Okay, so this one is peritubular, and this is how the things are getting here selectively reabsorbed. So this one is selective reabsorption, the process of selective reabsorption. Okay, when important substances like glucose, like protein, like amino acid, which should not be like excreted in urine. Okay, whatever is coming in this glomerular capsule, it is going into urine. Okay, whatever coming in this renal tubule, it is going into urine, and whatever coming into this peritubular capillary, it is going into circulation. So of course, what we need to give into the circulation, we will add in this peritubular capillaries, and what we need not to give into the circulation, we will discard into the urine. So this is what the theme is. Okay, so ultrafiltration or glomerular filtration is the thing. You can say this process of ultrafiltration. Another one is selective reabsorption, and the last one is this one is tubular secretion. Okay, so the substances, substances like toxic waste. Okay, toxic waste are toxins. You can say drugs. Okay, you can say creatinine. Okay, you can say it para-hippuric acid. Okay, so these are the toxic waste which is like not at all should be given into the circulation, rather it should be given into the urine, discarded out of the body, and this is how tubular secretion is occurring. And this peritubular capillaries are what? They are releasing, like they are secreting this inside what? They are secreting this inside the renal tubule so that it should be discarded out in urine. So this is how the three steps of uh, urine formation, you can say physiology of urine formation. In next class, we will discuss this in bit detail about the glomerular filtrate, what all are the factors affecting it, and what is selective reabsorption, and what is tubular secretion. I hope you understand, and let's meet in another video regarding the same thing. And till then,